bacteria are the oldest living organisms on the Earth. They've been here for billions of years. And what they are single-celled microscopic organisms. So they're one cell, and they have the special property that they only have one piece of DNA. So they have very few genes and genetic information to encode all of the traits that they carry out. And the way bacteria make a living is that they consume nutrients from the environment, they grow to twice their size, they cut themselves down in the middle, and one cell becomes two, and so on and so on. So they just grow and divide and grow and divide. So kind of boring life, except that what I would argue is that you have an amazing interaction with these critters. I know you guys think of yourself as humans, and this is sort of how I think of you. And so this man is supposed to represent a generic human being, and all of the circles in that man are all of the cells that make up your body. So there's about 1 trillion human cells that make each one of us who we are and able to do all the things that we do. But you have 10 trillion bacterial cells in you or on you at any moment in your life. So 10 times more bacterial cells than human cells on a human being. And so, of course, it's the DNA that counts. So here's all the A, T, G's, and C's that make up your genetic code and give you all your charming characteristics. So you have about 30,000 genes. Well, it turns out you have 100 times more bacterial genes playing a role in you or on you all of your life. And so at the best, you're 10% human, or more likely about 1% human, depending on which of these metrics you like. So I know you think of yourself as human beings, but I think of you as 90 or 99% bacterial. And these bacteria are not passive riders. These are incredibly important. They keep us alive. They cover us in an invisible body armor that keeps environmental insults out so that we stay healthy. They digest our food. They make our vitamins. They actually educate your immune system to keep bad microbes out. So they do all these amazing things that help us and are vital for keeping us alive. And they never get any press for that. But they get a lot of press because they do a lot of terrible things as well. So there's all kinds of bacteria on the Earth that have no business being in you or on you at any time. And if they are, they make you incredibly sick. And so the question from my lab is whether you want to think about all the good things that bacteria do or all the bad things that bacteria do. The question we had is how could they do anything at all? I mean, they're incredibly small. You have to have a microscope to see one. They live this sort of boring life where they grow and divide, and they've always been considered to be these asocial reclusive organisms. And so it seemed to us that they're just too small to have an impact on the environment if they simply act as individuals. And so we wanted to think if there couldn't be a different way that bacteria live. And the clue to this came from another marine bacterium, and it's a bacterium called Vibrio fischeri. And so what you're looking at on this slide is just a person from my lab holding a flask of a liquid culture of a bacterium, a harmless, beautiful bacterium that comes from the ocean named Vibrio fischeri. And this bacterium has the special property that it makes light. So it makes bioluminescence, like fireflies make light. So we're not doing anything to the cells here. We just took the picture by turning the lights off in the room, and this is what we see. And what was actually interesting to us was not that the bacteria made light, but when the bacteria made light. What we noticed is when the bacteria were alone, so when they were in dilute suspension, they made no light. But when they grew to a certain cell number, all the bacteria turned on light simultaneously. And so the question that we had is how can bacteria, these primitive organisms, tell the difference from times when they're alone and times when they're in a community and then all do something together? And what we figured out is that the way that they do that is that they talk to each other, and they talk with a chemical language. So this is now supposed to be my bacterial cell. When it's alone, it doesn't make any light. But what it does do is to make and secrete small molecules that you can think of like hormones. And these are the red triangles. And when the bacteria is alone, the molecules just float away, and so no light. But when the bacteria grow and double, and they're all participating in making these molecules, the extracellular amount of that molecule increases in proportion to cell number. And when the molecule hits a certain amount that tells the bacteria how many neighbors they are, they recognize that molecule, and all of the bacteria turn on light in synchrony. And so that's how bioluminescence works. They're talking with these chemical words. And the reason that Vibrio fischeri is doing that comes from the biology. So again, another plug for the animals in the ocean. Vibrio fischeri lives in this squid. What you're looking at is the Hawaiian bobtail squid, and it's been turned on its back. And what I hope you can see are these two glowing lobes, and these house the Vibrio fischeri cells. They live in there at high cell number. That molecule is there, and they're making light. And the reason the squid is willing to put up with these shenanigans is because it wants that light. And so the way that this symbiosis works is that this little squid lives just off the coast of Hawaii, so just in sort of shallow, knee-deep water. And the squid is nocturnal. So during the day, it buries itself in the sand and sleeps. But then at night, it has to come out to hunt. And so on bright nights, when there's lots of starlight or moonlight, that light can penetrate the depth of the water the squid lives in, since it's just in those couple feet of water. And what the squid has developed is a shutter that can open and close over this specialized light organ housing the bacteria. And then it has detectors on its back, so it can sense how much starlight or moonlight is hitting its back, and it opens and closes the shutter, so the amount of light coming out of the bottom, which is made by the bacterium, exactly matches how much light hits the squid's back. So the squid doesn't make a shadow. So it actually uses the light from the bacteria to counter-illuminate itself in an anti-predation device. And so predators can't see its shadow, calculate its trajectory, and eat it. And so this is like the stealth bomber of the ocean. But then if you think about it, this squid has this terrible problem because it's got this dying, thick culture of bacteria, and it can't sustain that. And so what happens is every morning when the sun comes up, the squid goes back to sleep, it buries itself in the sand, and it's got a pump that's attached to its circadian rhythm. And when the sun comes up, it pumps out like 95% of the bacteria. And so now the bacteria are dilute, that little hormone molecule is gone, so they're not making light, but of course the squid doesn't care, it's asleep in the sand. And as the day goes by, the bacteria double, they release the molecule, and then light comes on at night exactly when the squid wants it. And so first we figured out how and this bacterium does this, but then we brought the tools of molecular biology to this to figure out really what's the mechanism. And what we found, so this is now supposed to be, again, my bacterial cell, is that Vibrio fischeri has a protein. That's the red box. It's an enzyme that makes that little hormone molecule, the red triangle. And then as the cells grow, they're all releasing that molecule into the environment, so there's lots of molecule there. And the bacteria also have a receptor on their cell surface that fits like a lock and key with that molecule. These are just like the receptors on the surfaces of your cells. And so when the molecule increases to a certain amount, which says something about the number of cells, it locks down into that receptor, and information comes into the cells that tells the cells to turn on this collective behavior of making light. And why this is interesting is because in the past decade, we have found that this is not just some anomaly of this ridiculous glow-in-the-dark bacterium that lives in the ocean. All bacteria have systems like this. So now what we understand is that all bacteria can talk to each other. They make chemical words, they recognize those words, and they turn on group behaviors that are only successful when all of the cells participate in unison. And so now we have a fancy name for this. We call it quorum sensing. They vote with these chemical votes, the vote gets counted, and then everybody responds to the vote. And what's important for today's talk is that we know that there are hundreds of behaviors that bacteria carry out in these collective fashions. But the one that's probably the most important to you is virulence. So it's not like a couple bacteria get in you and then they start secreting some toxins. You're enormous. That would have no effect on you. You're huge. But what they do, we now understand, is they get in you, they wait, they start growing, they count themselves with these little molecules, and they recognize when they have the right cell number that if all of the bacteria launch their virulence attack together, they're going to be successful at overcoming an enormous host. So bacteria always control pathogenicity with quorum sensing. And so that's how it works. We also then went to look at what are these molecules. So these were the red triangles on my slides before. And so this is the Vibrio fischeri molecule. This is the word that it talks with. And then we started to look at other bacteria, and these are just a smattering of the molecules that we've discovered. And what I hope you can see is that the molecules are related. So the left-hand part of the molecule is identical in every single species of bacteria. But the right-hand part of the molecule is a little bit different in every single species. And what that that does is to confer exquisite species specificities to these languages. So each molecule fits into its partner receptor and no other. So these are private, secret conversations. These conversations are for intra-species communication. Each bacteria uses a particular molecule, that's its language, that allows it to count its own siblings. And so once we got that far, we thought we were starting to understand that bacteria have these social behaviors, but what we were really thinking about is that most of the time bacteria don't live by themselves. They live in incredible mixtures with hundreds or thousands of other species of bacteria. And that's depicted on this slide. This is your skin. So this is just a picture, a micrograph of your skin anywhere on your body. It looks pretty much like this. And what I hope you can see is that there's all kinds of bacteria there. And so we started to think, if this really is about communication in bacteria, and it's about counting your neighbors, it's not enough to be able to only talk within your species. There has to be a way to take a census of the rest of the bacteria in the population. So we went back to molecular biology and started studying different bacteria. And what we found now is that, in fact, bacteria are multilingual. So they all have a species-specific system. They have a molecule that says me. But then running in parallel to that is a second system that we've discovered that's generic. So they have a second enzyme that makes a second signal, and it has its own receptor, and this molecule is the trade language of bacteria. It's used by all different bacteria, and it's the language of interspecies communication. And so what happens is that bacteria are able to count how many of me and how many of you, and they take that information inside and they decide what tasks to carry out depending on who's in the minority and who's in the majority of any given population. And so then again, we turned to chemistry, and we figured out what this generic molecule is. So that was the pink ovals on my last slide. This is it. It's a very small 5-carbon molecule. And what the important thing is that we learned is that every bacterium has exactly the same enzyme and makes exactly the same molecule. So they're all using this molecule for interspecies communication. So this is the bacterial Esperanto. And so once we got that far, we've started to learn that bacteria can talk to each other with this chemical language. But what we started to think is that maybe there's something practical that we can do here as well. So I've told you that bacteria do have all these social behaviors that they communicate with these molecules. And of course, I've also told you that one of the important things they do is to initiate pathogenicity using quorum sensing. So we thought, what if we made these bacteria so they can't talk or they can't hear? Couldn't these be new kinds of antibiotics? And of course, you've just heard and you already know that we're running out of antibiotics. Bacteria are incredibly multi-drug resistant right now, and that's because all of the antibiotics that we use kill bacteria. So they either pop the bacterial membrane, they make the bacterium so it can't replicate its DNA. We kill bacteria with traditional antibiotics, and that selects for resistant mutants. And so now, of course, we have this global problem in infectious diseases. So we thought, well, what if we could sort of do behavior modifications, just make these bacteria so they can't talk, they can't count, and they don't know to launch virulence. And so that's exactly what we've done, and we've sort of taken two strategies. The first one is we've targeted the intra-species communication system. So we've made molecules that look kind of like the real molecules, which you saw, but they're a little bit different. And so they lock into those receptors and they jam recognition of the real thing. And so by targeting the red system, what we are able to do is to make species-specific or disease-specific anti-quorum sensing molecules. We've also done the same thing with the pink system. We've taken that universal molecule and turned it around a little bit so that we've made antagonists of the interspecies communication system. And these, the hope is that these will be used as broad-spectrum antibiotics that work against all bacteria. And so to finish, I'll just show you the strategy. And this one, I'm just using the interspecies molecule, but the logic is exactly the same. So what you know is that when that bacterium gets into the animal, in this case, a mouse, it doesn't initiate virulence right away. It gets in, it starts growing, it starts secreting its quorum sensing molecules. It recognizes when it has enough bacteria that now they're going to launch their attack and the animal dies. And so what we've been able to do is to give these virulent infections, but we give them in conjunction with our anti-quorum sensing molecules. So these are molecules that look kind of like the real thing, but they're a little bit different, which I've depicted on this slide. And what we now know is that if we treat the animal with a pathogenic bacterium, a multi-drug resistant pathogenic bacterium, in the same time we give our anti-quorum sensing molecule, in fact, the animal lives. And so we think that this is the next generation of antibiotics, and it's going to get us around, at least initially, this big problem of resistance. So what I hope you think is that bacteria can talk to each other. They use chemicals as their words. They have an incredibly complicated chemical lexicon that we're just now starting to learn about. And of course, what that allows bacteria to do is to be multicellular, right? And so in the spirit of TED, they are doing things together because it makes a difference, right? So what happens is that bacteria have these collective behaviors, and they can carry out tasks that they could never accomplish if they simply acted as individuals. And what I would hope that I could further argue to you is that this is the invention of multicellularity. Bacteria have been on the earth for billions of years, humans, a couple 100,000. So we think bacteria made the rules for how multicellular organization works, and we think by studying bacteria, we're going to be able to have insight about multicellularity in the human body. So we know that the principles and the rules, if we can figure them out in these sort of primitive organisms, the hope is that they will be applied to other human diseases and human behaviors as well. I hope that what you've learned is that bacteria can distinguish self from other. So by using these two molecules, they can say me and they can say you. And again, of course, that's what we do both as in a molecular way and then also in an outward way. But I think about the molecular stuff. This is exactly what happens in your body. It's not like your heart cells and your kidney cells get all mixed up every day. And that's because there's all of this chemistry going on, these molecules that say who each of these groups of cells is and what their tasks should be. And so again, we think that bacteria invented that, and then you've just evolved a few more bells and whistles, but all of the ideas are in these simple systems that we can study. And then the final thing is, again, just to reiterate that there's this practical part, and so we've made these anti-quorum sensing molecules that are being developed as new kinds of therapeutics, but then to finish with a plug for all the good and miraculous bacteria that live on the earth, we've also made pro-quorum sensing molecules. So we've targeted those systems to make the molecules work better. And so remember, you have these 10 times or more bacterial cells in you or on you, keeping you healthy. What we're also trying to do is to beef up the conversation of the bacteria that live as mutualists with you in the hopes of making you more healthy, making those conversations better, so bacteria can do things that we want them to do better than they would be on their own.
are we? That is the big question. And essentially, we are just an upright, walking, big brain, super intelligent ape. This could be us. We belong to the family called the Hominidae. We are the species called Homo sapiens sapiens. And it's important to remember that, and in terms of our place in the world today and our future on planet Earth. We are one species of about 5,500 mammalian species that exist on planet Earth today, and that's just a tiny fraction of all species that have ever lived on the planet in past times. We're one species out of approximately, well, let's say at least 16 upright walking apes that have existed over the past 6 to 8 million years. But as far as we know, we're the only upright walking ape that exists on planet Earth today, except for the bonobos, and it's important to remember that because the bonobos are so human and they share 99% of their genes with us. And we share our origins with a handful of the living great apes. It's important to remember that we evolved. Now, I know that's a dirty word for some people, but we evolved from common ancestors with the gorillas, the chimpanzee, and also the bonobos. We have a common past and we have a common future. And it is important to remember that all of these great apes have come on as long and as an interesting evolutionary journey as we ourselves have today. And it's this journey that is of such interest to humanity. And it's this journey that has been the focus of the past 3 generations of my family as we've been in East Africa looking for the fossil remains of our ancestors to try and piece together our evolutionary past. And this is how we look for them. A group of dedicated young men and women walk very slowly across vast areas of Africa looking for small fragments of bone, fossil bone, that may be on the surface. And that's an example of what we may do as we walk across the landscape in northern Kenya looking for fossils. I doubt many of you in the audience can see the fossil that's in this picture, but if you look very carefully, there is a jaw, lower jaw, of a 4.1 million year old upright walking ape as it was found at Lake Turkana on the west side. It's extremely time-consuming, labor-intensive, and it is something that it's going to involve a lot more people to begin to piece together our past. We still really haven't got a very complete picture of it. When we find a fossil, we mark it. Today we've got great technology. We have GPS. We mark it with a GPS fix, and we also take a digital photograph of the specimen, so we could essentially put it back on the surface, exactly where we found it, and we can bring all this information into big GIS packages today. When we then find something very important, like the bones of a human ancestor, we begin to excavate it extremely carefully and slowly using dental picks and fine paint brushes, and all the sediment is then put through these screens, and where we go again through it very carefully looking for small bone fragments. It's then washed. And these things are so exciting. They are so often the only or the very first time that anybody has ever seen the remains. Here's a very special moment when my mother and myself were digging up some remains of human ancestors. And it is one of the most special things to ever do with your mother. And not many people can say that. But now let me take you back to Africa 2 million years ago. I'd just like to point out, if you look at the map of Africa, it does actually look like a hominid skull in its shape. Now we're going to go to the East African and the Rift Valley. It essentially runs up from the Gulf of Aden, or runs down to Lake Malawi. And the Rift Valley is a depression, it's a basin, and rivers flow down from the highlands into the basin carrying sediment, preserving the bones of animals that lived there. If you want to become a fossil, you actually need to die somewhere where your bones will be rapidly buried. You then hope that the earth moves in such a way as to bring the bones back up to the surface. And then you hope that one of us lot will walk around and find a small piece of you. Again. So it is absolutely surprising that we know as much as we do know today about our ancestors, because it's incredibly difficult, A, for these things to be preserved, and secondly, for them to have been brought back up to the surface. And we really have only spent 50 years looking for these remains and begin to actually piece together our evolutionary story. So let's go to Lake Turkana, which is one such lake basin in the very north of our country, Kenya. And if you look north here, there's a big river that flows into the lake that's been carrying sediment and preserving the remains of the animals that lived there. Fossil sites run up and down both lengths of that lake basin, which represents some 20,000 square miles. That's a huge job that we've got on our hands. 2 million years ago, at Lake Turkana, Homo erectus, one of our human ancestors, actually lived in this region. You can see some of the major fossil sites that we've been working in the north, but essentially 2 million years ago, Homo erectus, up in the far right corner, lived alongside 3 other species of human ancestor. And here is a skull of a Homo erectus, which I've just pulled off the shelf there. But it is not to say that being a single species on planet Earth is the norm. In fact, if you go back in time, it is the norm that there are multiple species of hominids or of human ancestors that coexist at any one time. Where did these things come from? That's what we're still trying to find answers to. And it is important to realize that there is diversity in all different species, and our ancestors are no exception. There's some reconstructions of some of the fossils that have been found from Lake Turkana. But I was very lucky to have been brought up in Kenya, essentially accompanying my parents to Lake Turkana in search of human remains. And we were able to dig up, when we got old enough, fossils such as this slender-snouted crocodile. And we dug up giant tortoises and elephants and things like that. But when I was 12, as I was in this picture, a very exciting expedition was in place on the west side when they found, essentially, the skeleton of this Homo erectus. I could relate to this Homo erectus skeleton very well because I was the same age that he was when he died. And I imagined him to be tall, dark-skinned. His brothers certainly were able to run long distances chasing prey, probably sweating heavily as they did so. He was very able to use stones effectively as tools. And this individual himself, this one that I'm holding up here, actually had a bad back. He'd probably had an injury as a child. He had a scoliosis and therefore must have been looked after quite carefully by other female and probably much smaller members of his family group to have got to where he did in life, age 12. Unfortunately for him, he fell into a swamp and couldn't get out. Essentially, his bones were rapidly buried and beautifully preserved, and he remained there until 1.6 million years later, when this very famous fossil hunter, Kamoya Kimeu, walked along a small hillside and found that small piece of his skull lying on the surface amongst the pebbles, recognized it as being hominid. It's actually this little piece up here on the top. Well, an excavation was begun immediately, and more little bits of skull started to be extracted from the sediment. And what was so fun about it was the skull pieces got closer and closer to the roots of the tree. And fairly recently the tree had grown up, but it had found that the skull had captured nice water in the hillside, and so it had decided to grow its roots in and around this, holding it in place and preventing it from washing away down the slope. we began to find limb bones, we found finger bones, the bones of the pelvis, vertebrae, ribs, collarbones, things that had never ever been seen before in Homo erectus. It was truly exciting. He had a body very similar to our own, and he was on the threshold of becoming human. Well, shortly afterwards, members of his species started to move northwards out of Africa, and you start to see fossils of Homo erectus in Georgia, China, and also in parts of Indonesia. So Homo erectus was the first human ancestor to leave Africa and begin its spread across the globe. Some exciting finds, again, as I mentioned, from Dmanisi in the Republic of Georgia, but also surprising finds from recently announced from the island of Flores in Indonesia, where a group of these human ancestors have been isolated and have become dwarfed, and they're only about a meter in height. But they lived only 18,000 years ago. And that is truly extraordinary to think about. Just to put this in terms of generations, because people do find it hard to think of time, Homo erectus left Africa 90,000 generations ago. We evolved essentially from an African stock, again, at about 200,000 years as a fully fledged us. And we only left Africa about 70,000 years ago. And until 30,000 years ago, at least 3 upright walking apes shared the planet Earth. The question now is, well, who are we? We're certainly a polluting, wasteful, aggressive species with a few nice things thrown in, perhaps. But for the most part, we're not particularly pleasant at all. We have a much larger brain than our ape ancestors. Is this a good evolutionary adaptation, or is it going to lead us to being the shortest-lived hominid species on planet Earth? And what is it that really makes us us? I think it's our collective intelligence. It's our ability to write things down, our language, and our consciousness. From very primitive beginnings, with a very crude toolkit of stones, we now have a very advanced toolkit, and our tool use has really reached unprecedented levels. We've got buggies to Mars, we've mapped the human genome, and recently even created synthetic life, thanks to Craig Venter. And we've also managed to communicate with people all over the world from extraordinary places, even from within an excavation in northern Kenya. We can talk to people about what we're doing. As Al Gore so clearly has reminded us, we have reached extraordinary numbers of people on this planet. Human ancestors really only survive on planet Earth, if you look at the fossil record, for about, on average, 1 million years at a time. We've only been around for the past 200,000 years as a species, yet we've reached a population of more than 6.5 billion people. And last year, our population grew by 80 million. I mean, these are extraordinary numbers. You can see here, again, taken from Al Gore's book, but what's happened is our technology has removed the checks and balances on our population growth. We have to control our numbers, and I think this is as important as anything else that's being done in the world today, but we have to control our numbers because we can't really hold it together as a species. My father so appropriately put it that we are certainly the only animal that makes conscious choices that are bad for our survival as a species. Can we hold it together? It's important to remember that we all evolved in Africa. We all have an African origin. We have a common past and we share a common future. Evolutionary speaking, we're just a blip. We're sitting on the edge of a precipice. And we have the tools and the technology at our hands to communicate what needs to be done to hold it together today. We could tell every single human being out there if we really wanted to, but will we do that? Or will we just let nature take its course? Well, to end on a very positive note, I think evolutionary speaking, this is probably a fairly good thing in the end. I'll leave it at that. Thank you very much.
I have 18 minutes to tell you what happened over the past 6 million years. All right. We all have come from a long way here in Africa and converged in this region of Africa, which is a place where 90% of our evolutionary process took place. And I say that it's not because I'm African, but it's in Africa that you find the earliest evidence for human ancestors, upright walking traces, even the first technology in the form of stone tools. So we all are Africans, and welcome home. All right. I'm a paleoanthropologist, and my job is to define man's place in nature and explore what makes us human. And today, I will use Salam, the earliest child ever discovered, to tell you a story of all of us. Salam is an almost complete skeleton of a 3-year-old girl who lived and died 3.3 million years ago. She belongs to the species known as Australopithecus afarensis. You don't need to remember that. It's the Lucy species, and was found by my research team in December 2000 in an area called Dikika. It's in the northeastern part of Ethiopia. And Salam means peace in many Ethiopian languages. We use that name to celebrate peace in the region and in the planet. And the fact that we cover a story of all these famous magazines gives you already an idea of her significance, I think. After I was invited by TED, I did some digging, because that's what we do, to know about my host. You don't just jump into an invitation. And I learned that the first technology appeared in the form of stone tools 2.6 million years ago. First entertainment comes evidence from flutes that are 35,000 years ago, and evidence for first design comes 75,000 years old beads. And you can do the same with your genes and track them back in time. And DNA analysis of living humans and chimpanzees teaches us today that we diverged sometime around 7 million years ago, and that these two species share over 98% of the same genetic material. I think knowing this is very useful context within which you can think of our ancestry. However, DNA analysis informs us only about the beginning and the end, not telling us nothing about what happened in the middle. So for us, paleoanthropologists, our job is to find the hard evidence, the fossil evidence, to fill in this gap and see the different stages of development. Because it's only when you do that, then you can talk about it's only when you do that you can talk about how we looked like and how we behaved at different times, and how those likes, looks, and behaviors changed through time. That then gives you an access to explore the mechanisms, the biological mechanisms and forces that are responsible for this gradual change that made us what we are today. But finding the hard evidence is a very complicated endeavor. It's a systematic and scientific approach which takes you to places that are remote, hot, hostile, and often with no access. Just to give you an example, when I went to Dikika, where Salam was found in 1999, and it's about 500 kilometers from Addis Ababa, the capital of Ethiopia, it took us only 7 hours to do the first 470 kilometers of the 500, but took 4 solid hours to do the last only 30 kilometers. With the help of the locals and using just shovels and picks, I made my way. I was the first person to actually drive a car to the spot. When you get there, this is what you see, and it's the vastness of the place which makes you feel helpless and vulnerable. And once you made it there, the big question is where to start. And you find nothing for years and years. When I go to places like this, which are paleontological sites, it's like going to a game park, an extinct game park. But what you find are not the human remains, such as Salam and Lucy, on a day-to-day basis. You find elephants, rhinos, monkeys, pigs, etc. But you could ask, how could these large mammals live in this desert environment? Of course, they cannot, but are telling you already that the environment and the carrying capacity of this region was drastically different from what we have today. A very important environmental lesson could be learned from this. Anyway, once you made it, then it's a game park, as I said, an extinct game park, and our ancestors lived in that game park, but were just the minorities. They were not as successful and as widespread as the Homo sapiens that we are. To tell you just an example, an anecdote about the rarity, I was going to this place every year and would do field work here. And the assistants, of course, helped me and do the survey. They would find a bone and tell me, here is what you're looking for. I would say, no, that's an elephant. Again, another one, it's a monkey, it's a pig, etc. So one of my assistants, who never went to school, said to me, listen, Zara, you either don't know what you're looking for or you're looking in the wrong place, he said. And I said, why? Because there were elephants and lions and the people were scared and go somewhere else. Let's go somewhere else. Well, he was very tired, and it's really tiring. It was then after such hard work and many frustrating years that we found Salam, and you see the face here covered by sandstone, and here is actually the spinal column and the whole torso encased in a sandstone block because she was buried by a river. What you have here seems to be nothing but contains an incredible amount of scientific information that helps us explore what makes us human. This is the earliest and most complete juvenile human ancestor ever found in the history of paleoanthropology, an amazing piece of our long, long history. There were these three people and me, and I'm taking the picture, that's why I'm not in. How would you feel if you were me? You have something extraordinary in your hand, but you are in the middle of nowhere. The feeling I had was a deep and quiet happiness and excitement, of course accompanied by a huge sense of responsibility of making sure everything is safe. Here is a close-up of the fossil after 5 years of cleaning, preparation, and description, which was very long, and as I had to expose the bones from the sandstone block I just showed you in the previous slide. It took 5 years. In a way, this was like the second birth for the child after 3.3 million years, but the labor was very long. And here is, for scale, it's a tiny bone. And in the middle is the Minister of Ethiopian Tourism, who came to visit as a National Museum of Ethiopia while I was working there. And you see me worried and trying to protect my child, because you don't believe anyone with this kind of child, even a minister. So then, once you've done that, next stage is to know what it is. And to know, once that was done, then it was possible to compare. We were able to tell that she belonged to the human family tree because the legs, the foot, and some features clearly showed that she walked upright. And upright walking is a hallmark in humanity. But in addition, if you compare the skull with comparably aged chimpanzee and little George Bush here, you see that you have vertical forehead. And you see that in humans, because of the development of the prefrontal cortex, it's called, you don't see that in chimpanzees. And you don't see this very projecting canine. So she belongs to our family tree. But within that, of course, you do detailed analysis, and we know now that she belongs to the Lucy species known as Australopithecus afarensis. Then, next exciting question is girl or boy? And how old was she when she died? You can determine the age, the sex of the individual based on the size of the teeth. How? You know, in primates, there is this phenomenon called sexual dimorphism, which simply means males are larger than females and males have larger teeth than the females. But to do that, you need the permanent dentition, which you don't see here, because what you have here are the baby teeth. But using the CT scanning technology, which is normally used for medical purposes, you can go deep into the mouth and come up with this beautiful image showing you both the baby teeth here and the still growing adult teeth here. So when you measure those teeth, it was clear that she turned out to be a girl with very small canine tooth. And to know how old she was when she died, what you do is you do an informed estimate, and you say how much time would be required to form this amount of teeth, and the answer was 3. So this girl died when she was about 3.3 million years ago. So with all that information, the big question is, so what do we actually, what does she tell us? To answer this question, we can phrase another question. What do we actually know about our ancestors? We want to know how they looked like, how they behaved, how they walked around, and how they lived and grew up. And among the answers that you can get from this skeleton are included first, this skeleton documents for the first time how infants looked over 3 million years ago. And second, she tells us that she walked upright, but had some adaptation for tree climbing. And more interesting, however, is the brain in this child was still growing. At age 3, if you have a still growing brain, it's a human behavior. In chimps, by age 3, the brain is formed over 90%. That's why they can cope up with their environment very easily after birth, faster than us anyway. But in humans, we continue to grow our brains. That's why we need care from our parents. But that care means also you learn, you spend more time with your parents. And that's very characteristic of humans, and it's called childhood, which is this extended dependence of human children on their family or parents. So the still growing brain in this individual tells us that childhood, which requires an incredible social organization, a very complex social organization, emerged over 3 million years ago. So by being at the cusp of our evolutionary history, Salam unites us all and gives us a unique account on what makes us human. But not everything was human, and I will give you a very exciting example. This is called the hyoid bone. It's a bone which is right here. It supports your tongue from behind. It's, in a way, your voice box. It determines the type of voice you produce. It was not known in the fossil record, and we have it in this skeleton. When we did the analysis of this bone, it was clear that it looked very chimp-like, chimpanzee-like. So if you were there 3.3 million years ago to hear when this girl was crying out for her mother, she would have sounded more like a chimpanzee than a human. Maybe you're wondering, so you see this ape feature, human feature, ape feature. What does that tell us? You know, that is very exciting for us because it demonstrates that things were changing slowly and progressively, and that evolution is in the making. To summarize the significance of this fossil, we can say the following. Up to now, the knowledge that we had about our ancestors came essentially from adult individuals because the baby fossils were missing. They don't preserve well, as you know. So the knowledge that we had about our ancestors on how they looked and they looked like, how they behaved was kind of biased towards adults. Imagine somebody coming from Mars, and his job is to report on the type of people occupying our planet Earth, and you hide all the babies as the children, and he goes back, reports. Can you imagine how much biased his report would be? That's what somehow we were doing so far in the absence of the fossil children. So I think the new fossil fixes this problem. So I think the most important question at the end is, what do we actually learn from specimens like this and from our past in general? Of course, in addition to extracting this huge amount of scientific information as to what makes us human. You know, the many human ancestors that have existed over the past 6 million years, and there are more than 10, they did not have the knowledge, the technology, and sophistication that we Homo sapiens have today. But if this species, ancient species, would travel in time and see us today, they would very much be very proud of their legacy because they became the ancestors of the most successful species in the universe. And they were probably not aware of this future legacy, but they did great. Now the question is, we Homo sapiens today are in a position to decide about the future of our planet, possibly more. So the question is, are we up to the challenge? And can we really do better than this primitive, small-brained ancestors? Among the most pressing challenges that our species is faced with today are the chronic problems of Africa. Needless to list them here, and there are more competent people to talk about this. Still, in my opinion, we have two choices. One is to continue to see a poor, ill, crying Africa carrying guns that depends on other people forever, or to promote an Africa which is confident, peaceful, independent, but cognizant of its huge problems and great values at the same time. I am for the second option, and I'm sure many of you are. And the key is to promote a positive African attitude towards Africa. It's because we Africans concentrate, I'm from Ethiopia, by the way, we concentrate too much on how we are seen from elsewhere or from outside. I think it's important to promote in a more positive way on how we see ourselves. That's what I call positive African attitude. So finally, I would like to say, so let's help Africa walk upright and forward, then we all can be proud of our future legacy as a species. Thank you.
At the start of the dinosaur era, the continents are clustered together into a single landmass we call Pangaea. But now, they start to break apart. Africa separates from South America. The vast Atlantic Ocean opens up, creating what will become one of the defining barriers of human history, the gulf between the old and new worlds. The undisputed stars of the dinosaur era are animals like Triceratops and T-Rex. But there are some important creatures scurrying around their feet.
If we were to trace our lineage back far enough, we would come to really small shrew-like mammals surrounded by these titans of reptile life. "65 million years ago, a 6-mile-wide object, likely an asteroid, slams into the Earth. A dust cloud blocks out the sun. Temperatures plummet. Every creature on land weighing over 50 pounds goes extinct. The reign of the dinosaurs is over." The greatest gift that the dinosaurs ever gave us was dying. When they went extinct, it gave the mammals time to rise.
It doesn't take long after the disappearance of the dinosaurs for the first true primates to appear. Like their later versions, including us, these mammals have evolved forward-facing eyes, allowing for accurate depth perception and flexible hands with 5 digits.
They have 5 fingers, just like us, which means we can grasp things. If you think about other animals that don't have digits organized the way ours are, their ability to hold things, to manipulate objects, is much more limited.
50 million years ago, our primate ancestors are evolving on a planet that is warming. It's so hot, there are jungles at the poles. As the continents drift, the Americas and Africa have almost fully taken shape. But in northern Africa, modern-day Egypt is submerged beneath an ancient sea. On the floor of that sea live small-shelled creatures called nummulites. Their shells, made of calcium and carbon, pile up on the sea bottom over millions of years, where they form into limestone. Limestone that will be used to build the Great Pyramids. If you look closely at the pyramids today, you can still see evidence that these 4,000-year-old monuments are, in fact, made of 50-million-year-old seashells. By 10 million years ago, Earth is morphing into a world most of us would recognize. The Colorado River is carving out the Grand Canyon. Mountain ranges like the Himalayas have arisen. They're so tall, they disrupt weather patterns, setting the stage for a colder planet. The Isthmus of Panama emerges to connect North and South America, cleaving the connection between the Atlantic and the Pacific, disrupting ocean currents and tipping the world even more towards an ice age. With the planet getting colder, our primate ancestors hang on in the tropics. But a new creature is coming in that threatens to destroy them. 7 million years ago, our primate ancestors lived safely in the trees. But their neighborhood is about to be invaded. This newcomer will have as profound an effect on human history as any other living thing on Earth. It seems almost impossible to believe, but one of the most important things that will lead to the emergence of us is the emergence of grass.
The grasslands appear almost simultaneously around the world. We get the African savannas. We get the Eurasian steppelands. We get the North American prairies. We get the great grasslands of Argentina, appearing simultaneously around the world.
In eastern Africa, grasslands invade the traditional woodland habitat of our ape ancestors. With fewer trees and greater gaps between them, our ancestors have to adapt.
Apes would notice that there's more and more apes in the same tree and less food, increasing incentives for apes to go from one patch of food to a different one separated by grasslands. Now, one way to do it is to run like hell. The other way to do it is to extend one's food sources into the grasslands and seek out the foods that are available there.
And so, some apes make the move down into this stark new habitat. It's a landscape better suited to primates that can walk on two legs, keeping their heads up above the tall grasses to watch for predators. Standing on 2 feet is a revolutionary advance because it frees up our hands. Hands we will need to shape human history. 2.6 million years ago, early proto-humans, or hominids, walk an Earth whose rocks are loaded with the element silicon. Created in the cores of stars billions of years before, silicon is the second most abundant element in Earth's crust. One of its chemical quirks is the ability to bond with oxygen to form crystals that combine into solid rocks. Rocks that can be chipped and shaped without shattering.
Hominids started doing this 2.6 million years ago, breaking cryptocrystalline silicates to make sharp edges, and people use them for millions, literally 2.6 million years.
Simply having a modified stone with a sharp edge on it Now suddenly you have a hammer. You have a crude cutting edge. A simple modified stone means a human can suddenly do 1,000 more things than we could do previously. That little extra bit of technology enabled our ancestors to persist and eventually turn into us.
Silicon launches the first technological revolution, the Stone Age. Millions of years after it powers our first handheld devices, another chemical quirk of silicon will make it the height of technology once again. The next leap towards becoming truly human relies on a little-known secret of our home planet. In the known universe, it turns out Earth may have a rare and special power.
Of all the planets and moons in the solar system, we think that Earth is unique in the ability to sustain fire.
Other planets and moons have lightning and lava. But only on Earth do we have the two critical things we need for fire to burn. A vast fuel supply in the form of plants and trees, and an atmosphere full of oxygen to fan the flames.
If fire wasn't a possibility, you'd have nothing like us running around. Homo sapiens, they made a world with fire.
Our ancestors have fire firmly under control by 800,000 years ago. It's a skill that connects us back to the very beginning. Remember that all energy was created in the Big Bang, and all life is in a competition for our share of this energy. Using fire to cook is like having an external stomach to break down foods, releasing more calories, giving us more energy, which in turn allows us to support bigger brains. Fire is also the ultimate gateway technology. We will soon use it to turn clay into pottery, metal into weapons, water into steam power.
If you don't have fire, you can't have an internal combustion engine. No fire, no metal. No fire, no rubber. It's a technology that opens a world of possibilities for creatures that know how to use it.
200,000 years ago, the modern human has fully taken shape. The larynx, or voice box, which is high up in the throat in our ancestors, descends. More complex sounds are now possible. We begin to speak. For the first time, information can be shared between individuals and across generations.